We start this topic by recombinant DNA technology. It is really the art of taking a desired DNA segment, preparing it properly, and placing it into a suitable carrier so the cell can make use of it. The logic is simple. Cut the DNA carefully, join it correctly, deliver it into the host, and then let the host machinery do the work. Once that sequence is clear, the entire topic becomes a chain of very logical steps rather than isolated facts. The whole process follows a fixed pipeline: isolate, cut, amplify, insert, culture, and extract. That order matters because each step prepares the material for the next one. Many questions simply ask you to arrange these stages correctly, so the sequence itself is a high yield memory point. If this flow is firm in your head, the rest of RDNA technology becomes much easier to organize. The first step is breaking open the source cell and purifying the genetic material. Different cells need different wall-breaking enzymes: lysozyme for bacteria, cellulase for plants, and chitinase for fungi. After that, the unwanted contaminants are removed, especially RNA and proteins using ribonuclease and protease. The important exam trap is ethanol precipitation. DNA comes out only after chilled ethanol is added, so that small word chilled matters. Once the DNA is purified, it has to be physically collected, and that is where spooling comes in. The DNA strands appear as fine threads in suspension and can be wound around a glass rod. It is a very visual step, but conceptually, it simply means recovering purified DNA from the solution. A useful memory image is spinning spaghetti around a fork because that matches the way the strands are gathered. The cutting step is all about precision. Restriction endonucleases cut DNA at specific recognition sites, which is why they are the molecular scissors of this technique. The diagram also reminds you that DNA is negatively charged, so during electrophoresis, it moves toward the positive electrode, the anode. That is a classic trap. Many students confuse the directions, but the logic is straightforward once charge is remembered. PCR is the amplification stage, and it is one of the most important tools in modern biotechnology. The cycle has 3 steps: denaturation, primer annealing, and extension. In other words, the DNA is separated, primers bind, and polymerase builds the new strands. The real power of PCR is amplification from a tiny starting amount to millions or even billions of copies, which is why it is so central to cloning workflows. The enzyme that makes PCR possible is Taq polymerase, which comes from Thermus aquaticus. That origin is worth remembering because it is a guaranteed matching style exam point. Since this bacterium lives in very hot conditions, its DNA polymerase can tolerate the high temperatures used during denaturation. So the lesson here is not just the enzyme name, but the reason it works so well in PCR. Ligation is where the molecular pieces are sealed together. The source DNA, the vector DNA, and DNA ligase come together to form recombinant DNA. The key idea is that both the insert and the vector should be cut with the same restriction enzyme so that the ends match properly. This is one of those steps where accuracy matters more than speed because a poor match will not give a stable recombinant molecule. After the recombinant DNA is prepared, it has to be inserted into the host cell using different delivery methods depending on the target. Bacteria often use heat shock, animal cells may use microinjection, plant cells commonly use biolistics or gene gun, and disarmed pathogens can also act as vectors. The exam trick is to match the method with the organism: gene gun for plants, microinjection for animals. That association is much safer than trying to remember each one separately. Tell us whether the insertion worked. The slide uses ampicillin resistance as the marker, which is a very common screening idea in recombinant DNA work. Cells that did not take up the recombinant plasmid die in the presence of ampicillin, while transformed cells survive and grow. This is an important concept because selection is based on survival, not on direct visual identification. Bioreactors are the production stage, where the transformed cells are grown at scale under controlled conditions. The large stirred tank systems provide the right temperature, pH, oxygen, substrate, salts, and vitamins needed for high yield. Whether the vessel is simple or sparged, the principle is the same. Create an environment where the cells can produce the desired product efficiently. This is the bridge from lab-scale cloning to industrial biotechnology. Continuous culture is about maintaining cells in the active log phase for maximum output. Fresh medium is constantly added while used medium is removed, so the cells stay physiologically active instead of entering a stationary phase. That is why this system is so effective for large-scale recombinant protein production. A simple way to remember it is to think of it as a constantly refreshed system that keeps the culture productive for longer. The product is not finished when it leaves the bioreactor. Downstream processing is what makes it usable. This includes biosynthesis completion, separation and purification, formulation, clinical testing and quality control, and finally, market-ready product preparation. The important idea is that purification is not an optional extra. It is part of making the product safe, stable, and effective. In many questions, this stage is overlooked, so it helps to remember that the actual product becomes ready only after downstream processing. This summary slide is the fastest way to revise the entire sequence. Enzymes handle the key actions: lysozyme, cellulase, and chitinase for lysis, restriction endonucleases for cutting, DNA ligase for joining, and Taq polymerase for amplification. The process flow goes from isolation to gel electrophoresis, PCR, insertion, culturing, and downstream processing, while bioreactors support active growth. If you can recite this blueprint without hesitation, the topic is in strong shape for the exam. The final checkpoint is about confidence and recall. Recombinant DNA technology is really a sequence of controlled actions. Isolate the DNA, cut it precisely, amplify the desired segment, insert it into a vector, grow it in the host, and purify the final product. The exam usually rewards students who remember both the order and the exceptions, so focus on enzymes, delivery methods, selectable markers, and downstream processing. If those links are clear, you are ready to handle the chapter with confidence.